from the notes. Okay, it show about this one. Okay, so Monty, first you can go to this part first. Okay, okay, I just send you a whole picture. Okay, you can see my screen now. I'm showing the slide now. Okay?
No.
I haven't. Sorry. Okay, this one. Okay, Monty.
Yeah, I can see it now.
Yeah. So Monty. So in general, okay, you can have a read like what kind of question like to come out for these chapters. So okay, have a read, and then you should know how to describe what is meant by the physical digestions and what is meant by the chemical digestions. Okay, so this is thing that you need to know. You need to know how to describe it. Okay, and then okay, so this is about the balanced diet now. Okay, which is the diet that consists all kind of nutrients in correct proportions. Correct proportions, understand? You know what's meant by correct proportions? Which is you cannot be too much or too less.
Yeah.
Yeah, okay. And then the functions for each nutrient, right? So the carbohydrate is being used for what? Carbohydrate is being used in the respirations, which is usually in the aerobic respirations. So this one you can release energy to your body itself, right?
Yeah.
Yeah, and then so that's why carbohydrate is the primary source of energy. You agree with me?
Yeah.
Yeah. So that's why, Monty, you eat bread, you eat rice every day. You cannot survive without rice, right?
Yeah.
Yeah, okay. And then you should know about the fat and oil, okay? And then the fats and oil, right? This is like, okay, they will help in what inflammation and that kind of thing. And for the protein, right? Okay, protein, right, is usually for the growth and repair of tissue. So means that whenever you have an injury to your muscle or to your organs, so your body will repair it by producing the proteins. Because example of protein product is what? Enzyme, hemoglobins. Okay?
Okay.
Yeah, and also vitamin C. Vitamin C, right, they will help in to make the collagen. So means that, Monty, if you are lacking in the vitamin C, you will have scurvy. Scurvy, which is your gum will bleeding.
Okay.
Yeah, because vitamin C help to make out the collagens. You know your skin. You know your skin is made from collagens. Your face also got the collagen fibers, so you look young.
Okay.
Yeah, so that's why if you're lacking of collagen fibers, your skin, right, will be less elastic, and then your skin or your tissue, right, will be more easy to get bleeding. Understand? Because it's not strong. Then for the vitamin D, right? Vitamin D, okay, what they do is they will help to absorb the calcium for the strong bone and teeth. This is their functions. Absorb the calcium means that so this means that if you only have calcium but without the vitamin D, what will happen to you? Your calcium will still remain in blood, cannot uptake into the bone and teeth. Understand?
Okay.
So that's why there's importance for you to have vitamin D. And then you can get vitamin D from where? Sunlight.
Yeah.
Yeah, so vitamin D is to absorb calcium. Okay, the keyword, Monty, take note about keyword. Absorb calcium, okay, for the strong bone and teeth. Absorb calcium for the strong bone and teeth, okay?
Okay.
Yeah, and then for the calcium, right, is help in the blood clotting. And example is a ricket. Okay, it's a ricket, which is if you are lacking of the calcium, your bone will be very, very soft. So called soft bone. The condition known as ricket. Okay, and then for the ions, right, iron is like components to make hemoglobin. So hemoglobin is used to make your red blood cell, right? Your red blood cell is made for the hemoglobin. So it's a kind of protein. The hemoglobin that you are lacking of red blood cell also. So you have anemia. Okay, what's meant by anemia? You have a low count of red blood cells.
Okay.
Yeah. Okay, and then fibers, right? Fibers, they increase the bulk of your feces, allow your feces to increase in volume. So they will become softer and more moisture inside. So you can prevent constipation happen. Okay, prevent constipation happen.
Okay.
Yeah, and then the exam count before they ask you to describe about the vitamin D and calcium, the functions. Okay, the functions. Yeah, so this is the thing, right? Okay, vitamin D help to absorb calcium, prevent rickets, to build strong bone.
Okay.
Okay, and then aluminum canal, right? You just need to label. Okay, you just need to label. When you label, right, you just need to follow this kind of thing to label. Okay, let me show the reasons, the reason question. Okay. This one I also say in the group. Do you see this?
Yeah.
Yeah, okay. Yeah, I do say in the group, right? Okay, like this, okay. Yeah, I just need to label, okay, label for each the organs, label, okay. So that's why I think you already know, right? What is mean by L? What is L? L is the gallbladder.
Yeah. Okay.
Yeah, K is the liver. So you should know about the each structures. And then yeah, this I will not talk too much about it already because you already know. So you just need to know that the functions, right? The mouth, okay, mouth. What the mouth will do? The mouth, right? They usually will do they're able to do chemical digestions and physical digestions. And then physical digestion is usually by what? So your teeth and also stomach, right? Stomach.
Yeah.
Yeah, so they can churn the food. Yeah, so this one is physical. This is physical by the teeth, right? And then one more, emulsifications. Okay, Monty, emulsification is also example of physical. Yeah, okay. This one I will not talk about it again. So just need to know that liver is the one who produces bile. So bile is not an enzyme, you know. Bile is not enzyme. Bile cannot do chemical digestions. Okay.
Okay.
So what the bile can do? Tell me the function of bile, Monty.
It emulsifies fats and it keeps the pH neutral.
Yes, and then second functions.
It keeps the pH more neutral because it's an alkaline.
Okay, you should say that not keep the pH neutral. You should say that neutralize the acidic kind. Neutralize the acid.
Oh.
Okay, the things that you should write is you should write it as neutralize the acid. I think I could show. Yeah, see this one. Look at the second function for the bowel. Okay, neutralize the acidic kind from the stomach, so you can create the optimum pH in the small intestines.
Okay.
Yeah, so this is the keyword that you should write. So because the small intestines, the pH inside the small intestines, for example, in the duodenum, right, it's not totally neutral, Monty. The pH inside the duodenum is how much? Have a guess.
It's 4.
You can see from here. Here.
Oh. It's
yes, duodenum, the pH, the number beside, yeah.
8.
Yes, it's about 8 like that, 8 to 9 or to 10. So that's why this tell you what, Monty. This tell that your mouth, right? Your mouth is neutral, but when you go to the stomach, right? Stomach is acidic, okay? It's about pH 2 due to the presence of pepsins, right? And then when you go to the intestines, right? When you go to the intestines, for example, in the duodenum part, it'll be more to alkaline, more to alkaline. So that's why the enzyme which is what? The enzyme which is the trypsins and the lipase, right? Trypsin and lipase, which is acting in the duodenum, right? Okay, so you see these two enzymes. Trypsin is a kind of protein, lipase is a kind of enzyme, so they will just acting in the duodenum. So they are all working in the alkaline environments in the duodenum. Understand?
Yeah.
Same to the maltase. Okay, maltase also being produced in the small intestine. Yeah, so they're also acting in the small intestine, so they are more to alkaline. So just remember, inside the small intestine is more to alkaline.
Okay.
And then what the exam they would like to count? Exam they would like to count is like they ask you to compare. Okay, compare the pepsin and trypsin. And then how you able to differentiate them. So that's why Monty, pepsin and trypsin, right, is belong to which class of enzyme? They will digest what kind of molecule? What kind of molecule?
Protein.
Protein. So that's why they are proteins, right? So pepsin is at the stomach, trypsin is at small intestines. Yeah. So this is their pH range. Okay. Can you see the pH range?
Yeah.
Yeah. So that's why in the pepsin, right, it's more to pH 2. Okay. In the trypsin, it's more to pH 8. Okay.
Okay.
That's it. Yeah. Okay. So Monty, I hope that this table made you clear. Okay.
Okay.
Yeah, because right, the exam also come out in the same thing. They will ask you to fill up the enzyme, okay, and the substrate and the products, and then where is produced from. Yeah, where is it produced from? So you can see, right? Like in the amylase, okay, amylase is being produced from the salivary glands and pancreas. So and then they will be working, okay, they will be acting in the stuff in the mouth and in the duodenum. And then for the trypsin and lipase, right? So trypsin and lipase is being produced from the pancreas. So your pancreas then can produce the enzyme. Pancreas will produce the enzyme also. So after the pancreas produce the enzyme, so they will go out, okay, they will secrete out to the intestines. So they will perform the digestions in the intestines, which is the duodenum.
Oh, okay.
So I hope you are clear with me. And then okay, so we go back to have a look first. Okay, so that's why you should know about the whole process of the food digestions, which is from the ingestion, right? Ingestion to the digestions. So the digestion they go include the physical digestion. Okay, you can say mechanical or physical, but it's better if you write physical because the reference tables they change to the word physical.
Oh, okay.
But it's the same meaning, yeah. So the physical digestion involved in the chewing, churning action of stomach, and then the emulsifications, and then the chemical digestions all involve about enzyme only. Okay, about enzyme only. So that's why the nutrient can be absorbed by the villi and then go to the hepatic portal veins to the liver to do assimilations and ejection feces. So Monty, I think you are not clear about this one, right? This part you are not clear, right?
Yeah.
Yeah, okay, so let me go through with you what's meant by assimilations. Okay, so just imagine, right, Monty, once you swallow the food, right, the food will go to the esophagus and then go to stomach and then to intestines, and then they will carry out all the digestion process, right, the physical and the chemical. So that's why your food particle, right, will break down from large, become small particle, okay, right?
Okay, yeah.
So they will become small particles, and then they will become soluble also, all right, because they undergo chemical digestions.
Yeah.
Yeah, so that's why definition for the chemical reaction have a read, Monty. Yeah, it's a breakdown of the large insoluble molecule, right, into small and soluble molecule that can be absorbed.
Okay.
Yeah, so this is a definition, okay. Yeah. So that's why means that the food molecule after they undergo digestion process, right, from large they become small. So once they become a small molecule, they can be absorbed. Okay, now they can be absorbed to the where. So which is the site they will absorb the nutrient molecule? Where which is the organs?
Oh, can you repeat your question?
Okay. What is the organs which help to absorb the nutrients molecule?
Oh, the small intestine.
Yeah, the small intestine. So just remember that your small intestine they will just absorb the nutrient molecule. Right? Yeah,
Okay.
they will absorb the nutrient molecule. So that's why, yeah. So now the nutrient molecule, okay, which is the fatty acid and glycerol, right? Fatty acid glycerol from the lipid, and then yeah, because just imagine that the proteins, right? The proteins which is large and insoluble, they will break down by proteins, right? See, they just break down the proteins and then they'll become what? They become amino acids. Understand?
Okay.
Yeah, they become a small molecule which is amino acid, which is water soluble. Yeah, so amino acid, okay. Monty, I hope you still remember amino acid is a monomer, right? Which is the simplest form.
Yeah.
Yeah, so that's why it's very small inside, so they can directly absorb into the blood capillary in the villi.
Okay.
Okay, and then for the carbohydrates, right, which is the starch or glycogens, okay, usually starch, right, we talk about starch because we eat the food, right? So the starch will be break down by the amylase, become what? Maltose. Yeah, maltose is a disaccharide or polysaccharide or monosaccharide. Which one? Maltose.
It's a disaccharide.
Yes, okay. So disaccharide, can they directly absorb by the intestinal villi? Can they? Cannot.
No, not yet.
Because it's disaccharide. So that's why maltose they should need to further break down glucose. So maltose will break down by maltase. Maltase is an enzyme to break down the maltose become glucose, right? So that's why glucose is water soluble and then it's the simplest monomer unit. So they can uptake into the blood capillary in the villi.
Okay.
Okay, and then now we go to lipase. Okay, lipase. So that's why the fats, okay, the fats will be break down by lipase, become what? Become their monomer, which is a fatty acid glycerol. Okay. So fatty acid glycerol, they are what? They are lipid soluble. So that's why because they are lipid soluble, right, they cannot dissolve in the blood plasma. Okay, so Monty, why lipid soluble molecule cannot dissolve in blood plasma? Why? It's because they are lipid soluble.
Okay.
Just like you cannot dissolve the oil in the water, right? They will always separate. Correct.
Oh, yeah.
Yeah, so that's why lipid they cannot go into blood capillary because blood capillary inside they consist a lot of blood plasma. Blood plasma is made from water. Okay, half of the blood plasma is made from water. So if the fatty acid glycerol go to the blood capillary, right, it cannot be further transport. Understand? Because it cannot soluble in the water.
Okay.
So that's why they need to instead they go to lactule. Okay, they just go lactule in the bleeding. And then you should know where's the lactule, right? See, you should know about this one. Okay, you should know about structure of the intestine villus with the epithelial cell, right? So you can see the epithelial cell is just lining by the epithelial projections, which is a microvilli. Okay, you have one microvilli, which is here. Okay, see.
Okay.
See? Yeah. So this all thing, right? All these small finger-like projections, this one, they are all microvilli. This whole thing, right? This whole thing is a villi.
Okay.
Understand? Yeah, because microvilli is much more smaller. So microvilli is usually located on the surface of the villi, how to increase the surface area. And then you have a blood capillary. Okay, you have a blood capillary, and then so that's why the neutral molecule, which is a okay. So Monty, what kind of neutral molecule can go to the blood capillary?
Can you
Okay, what kind of neutral molecule they can directly absorb to the blood capillary? The glucose, okay? And then another thing is amino acid.
Oh, okay.
Yeah, so Monty, you can write down here, okay, blood capillary. So that's why glucose, okay, glucose and the amino acid is absorbed into here. And then for the lactule, right, it's a fatty acid. Okay, write down fatty acid. And glycerol, okay, you can write glycerol. And the glycerol. Glycerol. And also the fatty is, I mean the fatty tissue. Oh, I mean the fat soluble vitamin. Okay, so Monty, you should know that the glucose amino acid itself, right, is a water soluble molecule. It is water soluble, so they cannot dissolve in lactule. They only can go to the blood plasma, which is made from water also.
Okay.
Okay, so that's why this is this is a process, right? And then you should know about the pathway. Okay, the pathway, for example, right, like the after the fats, okay, they absorb to the lactule in the villi. So later from the lactule, right, they can go to the lymphatic vessel. Lymphatic vessel is another kind of vessel, okay, which is they made from a lot of lymphatic capillary. Yeah, so they just carry the lymph. They just carry the lymph fluid, yeah, because lymph fluid in the lymphatic system. But later, they'll just drain back this lymph fluid to the circulatory system.
Okay.
Yeah, which is the molecule, right? Okay, initially from the blood plasma, not the blood plasma, sorry. Initially from the intestines, right, and to the lactule. See, intestines to the lactule to the lymphatic system, back to the circulatory system. So in overall, this means that your nutrients can only absorb in form of the monosaccharide, which is a monomer.
Okay.
Yeah. Okay, so have a look on the functions yes.
Can you write it down?
Oh, you mean write down monomer, this one?
No, just now. What you said. Oh, okay.
Okay, yeah, this one, which is in form of monomer. Yeah.
Oh, like the smallest one, right?
Yeah. Smallest unit, yes, the smallest unit. So that's why, like the glucose, okay, monomer, for example, is what? Glucose, the amino acids, yeah, the fatty acid and glycerol. They are all from the simplest unit.
Okay.
So that's why the enzymes is trying to break them down.
Okay.
Okay, so I hope you're clear about the process, right? Okay, now we go to talk about the small intestine first.
Okay.
So Monty, here is a small intestine. So small intestine is a site which they will absorb the nutrients, right? They absorb the most of the nutrients, okay? Most of the molecule. Because small intestine, right, is longer. You know that small intestine is longer than your large intestines?
Yeah.
Yeah, so that's why most water is absorbed for the small intestines compared to the large intestines. Just like some of the water absorbed by the colon. The colon is in the large intestines.
Okay.
Because small intestines, they have a longer in their length than the large intestines.
Okay.
Okay, and then now we want to have a look on the structure of the vilus, right? So vilus, you have what? You have vilus and microvilus, okay? So why you need to have microvilus, Monty? Why you need to have microvilus?
It increases the surface area even more.
Yes. Yeah, so, and then what you can do, because as then they will ask you to explain it, yeah.
So it can absorb more nutrients and water.
Yes, okay, you can say in this way, okay, say like, you can increase the faster rate of absorption of the nutrient. Okay, or
Okay.
faster absorption of nutrient also can. And then why they need to have a very thin wall or one cell thick?
So that the nutrients can easily get into the villi.
Yeah, because you want to reduce the distance, right? You want to reduce the distance. So it's better for you to have a very thin wall. So that's why they can pass through them easily in a shorter distance, right?
Yeah.
Yeah, compared to a thick wall. Yeah. So thin wall is just like this, okay? Thin wall is just like this, right? They can pass through easily compared to thick wall. Thick wall is just like this. They need to travel long distance, which is not good for them.
Okay.
So it's one cell thick. One cell thick to ensure the short diffusion distance, okay? Short diffusion distance for the transport of nutrient, you can say it this way.
Okay.
And then this this intestine villi, right? They have a lot of good blood supply. See, there are a lot of good blood supply around them. Yeah, here, the good blood supply, and also they will have a mitochondria. Yeah, don't forget mitochondria. It means that you have a lot of mitochondria in your intestine villi, right? Which is your epithelial cell, right? So that's why the mitochondria, right? They will help to provide energy for the energy transport. What is meant by this one? Energy transport.
So. Okay.
Okay. What kind of nutrient can be actively transported by the villi into the blood capillary?
Can you repeat that?
Okay. What kind of nutrient can be actively transported by the intestine villi get into the blood capillary?
The glucose and amino acids.
Okay, only the glucose.
But like.
Yes.
But like you said those. Okay.
Okay, only the glucose are not amino acid. Yeah, because amino acid will be another way. Amino acid is by the diffusion.
Okay.
Amino acid is by diffusion, but the glucose, right? Glucose they can being absorbed. You know, Monty, glucose can absorb into the release. This one, glucose, right? Just imagine glucose, right? This one, okay, it's a 6 carbon molecule, right? They can uptake into the microvilli, okay, and get into the villus, right? By 2 methods. First is diffusion, another one is active transport.
Okay.
So there's 2 ways. Okay, so just remember that glucose can be transported by 2 ways into the microvilli.
Okay.
Diffusion and active transport. This is depend on their gradients.
Okay.
So that's why this is very important that your intestine villi, they should have a mitochondria. You should have mitochondria so you can provide energy for the active transport.
Okay.
And then you should have the blood capillary also. Because this blood capillary will help to transport the glucose and the amino acids away.
Okay.
And then you have a lactose. See, lactose help to transport what? Transport any kind of fatty acid, cholesterol, and fat soluble vitamins.
Okay.
Yeah, so this is the if they come in paper form, example question like this. Okay, so that's why for example, right? Okay, Monty, maybe I can show you questions about the nutrient transport pathway, right? Because our this note is just very complete already. You can just read this note and then you know how to answer it.
Okay.
Okay, so you see the question is just similar like this. See, it's very similar. Like the maltose is digested by maltase become what? Glucose, right? And then this process is happen in the epithelial lining of the intestines, right?
Okay.
And then like why the enzyme itself they cannot digest for example, like why the amylase, right? They only can digest starch in the stomach, but I mean, they only can digest starch in the small intestines, not in the stomach. It's because what you can say about pH. Because if in the stomach period, right, the stomach period is too acidic, so they will denature the amylase. I should write at this point, Monty. They should denature the amylase, and then they will change the shape of the active site.
Okay.
This is a key word. So that's why the active site is not able to bind to the substrate because it's no longer complementary shape already.
Okay.
So that's why this is your importance for the bile is being produced. So that's why the bile they can neutralize the acid to provide the suitable pH, but the pH is not neutral.
Okay.
It's more to alkaline. Yeah, it's more to like from 2 they will become 8 or 9 or 10.
Okay.
Is it overload or information, Monty? So far so good.
Yeah.
Yeah, I think you learned this before. Just like need to how to recap on it. For example, right, they will ask this kind of question. Let me show you about the questions. See, how about the balanced diet? Okay, the balanced diet, and what's the function of vitamin D? Yeah, okay, so this is a feeders, right? So they ask about the injection and then about the type of the two functions. So Monty, just need to know that your two function, right? You have what? You have the molar and premolar, so they will help you to in the chewing and grinding of food. You know what's meant by chewing and grinding? Like,
Yeah.
Like you're eating the food. Yeah. So this is the functions of the teeth, right? You should know about teeth structure and function. So and then this is the leaders. Okay. Yeah, see? They ask about what? Okay, they ask about where the villi is located. So it's in the small intestines.
Okay.
And function of lactose. Yeah, see the same question. And this one, describe the pathway taken by the product of protein digestion from villi. I mean, from this villi to the liver.
Okay.
Yeah, see, product of protein digestion from the villi to the liver. So that's why, Monty, you should know about the pathway. Okay, so that's why we go back to the story. Yeah, you can have a look on this, Monty. You can go to this page first.
Okay.
Go to the liver part. I go to open my aircon now. Wait for a while.
Oh.
Okay, Monty. Yeah, see about liver part. Okay, so now I want to discuss with you about liver functions. So what is the function of liver? Liver function, right? They will help in what? They will help in the metabolism, right? For example, they can help to convert the excess glucose in the blood become glycogen in the liver and muscle.
Okay.
Okay, you can follow, right? Okay, which when there's excess glucose in your blood, right? They can be converted by the insulin. So insulin they will become what? Glycogen, and then this glycogen is stored in the liver and muscle cell. Understand?
What's insulin?
Insulin is kind of hormones.
Oh.
Insulin, okay. So which is insulin? Yeah,
What
insulin.
do it?
Yes, hormones to reduce the blood glucose level. Reduce the blood glucose level. Okay.
Okay.
Yeah. And then liver. Okay, so that's why the importance of insulin. So Monty, I'm not sure. Have you had the kind of feeling that every time you're just having a heavy meal, right? And then after 1 hour, you feel very tired, want to sleep already.
Yeah.
Because of what? Because that after you have after you finish your heavy meal, you eat a lot of carbohydrates, right? So that's why the carbohydrate will break down by the enzyme, which is the amylase, right? Amylase, and then become the maltose, and later they become glucose. So that's why after you having your meal, your blood glucose level will be increasing, right?
Yeah.
And then your body will respond by release the insulin. So insulin is kind of hormones. So they're just trying to reduce out the glucose level. So how they reduce the blood glucose level? They will just convert the glucose become glycogens. Understand? They will just convert the glucose become glycogens.
Okay.
Glucose is just like this, Monty. Glucose to the glycogens, maybe you can see my notes, which is here, I think. Right, which is here. Chapter 4. Monty, you are not sure for some point? I feel like you are not sure for some point.
Yeah, I don't think I learned it before.
Oh, okay, it's okay. You learn in future, but I just want to let you know only, okay? Like this is the monosaccharide. You can see the left side, which is only the one sugar, right?
Yeah, I didn't know.
Glucose, okay. Then this is the starch. Like, can you see the starch? The structure of starch.
Yeah.
Yeah. So that's why your body, okay, they can store excess glucose. Yeah, they want to store excess glucose in form of the glycogens in your body, in the animal body. Understand?
Yeah.
Yeah, so that's why glycogen is what? Glycogen is the energy storage compound in human.
Okay.
Yeah, okay. This one, glycogen, right, is energy storage compound in human. I think your mom print out this note before, right? Yeah, this one.
Yeah, I think so.
Yeah. Okay, you can refresh that because it's like connected to each other, the knowledge. Yeah, so that's why if they convert the glucose become glycogens, and then since the glycogen is just very large and complex molecule, right? You can see the structure just now, very large, very complex, right?
Yeah.
Can store in the liver and the muscle. Okay, so this is about carbohydrate, right?
Okay.
Okay, so that's why this is the carbohydrate metabolism. Okay, so which is you can read like from the starch in the food, right? Monty, from the starch in food, they will be ingested by the mouth. So because you should know about pathway. So later the starch digestion will occur in the mouth, right? Yeah, starch digestion will occur in the mouth. So with the salivary amylase, so there will be convert become the maltose. Okay, maltose, right? So from the maltose, okay, we convert become the glucose at the end in the small intestines.
Okay.
Okay, this one you clear, right? I will just go down. So what happened if now the glucose, okay, so now the neutral molecule is become the glucose, which is simplest monomer unit, simplest form. Glucose is simplest form, right? Glucose.
Yeah.
Yeah, so that's why glucose can be directly absorbed by the small intestines, correct?
Yeah.
Yeah, and then since the glucose is the monomer, right? Since the glucose is just a monomer, so that's why they can directly go into the blood capillary.
Okay.
Yeah, so okay, later, Monty, you can see the knots, right? Okay. Later from the blood capillary in the small intestines, where they will go. The next step.
It'll go.
They will go to where?
Is it like it'll go to the liver and then the liver will perform assimilation?
Yes. Well done.
Okay.
And then Monty, tell me what is the name of blood vessel which help to transport the glucose from the small intestine to the liver? Tell me the name of the blood vessel, which is this one. Yeah, tell me.
The blood capillaries.
No blood capillary.
Oh,
Hepatic portal vein.
Oh.
Okay, so that's why this blood vessel, you can just write it down. You can write it down.
Okay.
Hepatic portal vein. This is very important. Hepatic portal vein. So hepatic portal vein is a kind of specialized blood vessel. It's a vein, right? It's a vein. So inside they carry a lot of deoxygenated blood, right? Deoxygenated blood. Deoxygenated blood. But it also carry a lot of nutrients. So it's a nutrient-rich molecule.
So it carries deoxygenated red blood cells and the nutrients.
Correct. Yes, nutrient. Okay, they carry what kind of nutrient? Tell me. They carry what kind of nutrient inside?
Glucose.
Yes, and also?
And amino acid.
Yes, these two things.
Okay.
So later, so this is the way that glucose, right, from the small intestines, they will just go to the liver. Yeah, and then liver, they will do the assimilations. Okay, Monty, tell me what's meant by assimilations. What's the definition for assimilations? Which is a nutrient molecule become part of the cell.
The nutrients are being moved around to the body to provide energy.
Okay, you just say that nutrient molecule become part of the cell. You can just refer to my definition at the keyword list.
Okay.
Yeah, okay. But what you say is not it's not wrong also. What you say is quite incorrect. Correct also, but it's better if you just follow the keyword, okay, which is here. Yeah, assimilation. Yeah, Monty, this is a what? See, the movement, okay, Monty, the movement of the digested food molecule into the cell or body where they are used becoming part of the cell.
Okay.
Okay, this one, yeah. This is definition if they ask you about this.
Can I write it down?
Yeah, you can write it down, okay. I think you might also print out this note, right? So you can copy down, yeah.
Yeah, okay. Oh, so they're being used as energy?
Yes, correct. Okay, Monty, you see the picture. I hope you love in this picture. I hope you feel in love with this picture. See, once the glucose, right, it enter to the liver. Okay, if there is excess glucose, if there's excess glucose, they will become glycogens, right?
Yeah.
They will become glycogen and stored in the liver. But what happens if there's no excess glucose, right? The glucose is just like sufficient. So usually the glucose, right, it will just go into every body cell. So this is the assimilation. Okay, they just go to the liver cell. Okay, they just go to the liver cell to become the aerobic respirations, or they go to other kind of body cell. Yeah, so that's why this is the one which is the nutrient molecule become part of the cell. Because like the body cell, they just uptake the glucose to release energy. So they can carry out the cellular aerobic respirations because they require glucose supply.
Okay.
Yeah, so this is the story about the carbohydrate, right? So you can just refer to the upside down. Yeah, see? So liver is assimilation, which is the nutrient molecule become part of the cell. So that's why there will be glucose uptake by the body cell for the cellular aerobic respirations, which is the equation. This is the equation. Yeah, so that's why if you have excess glucose in the blood, right, it will be converted by the insulin. Okay, insulin is a hormone. So this insulin, they will just convert to the glycogens in the liver. And also in the muscle cell. So this means that your body can store excess glucose in the form of glycogens.
Okay.
Okay, you're clear, right? You're clear, right? Yeah, the pathway. So just remember this picture only.
Yeah, okay.
Now we go to talk about proteins, right? Which is a bit confusing, proteins. Yeah, so that's why imagine, okay, Monty, have a look at this picture. See? Monty, you see? Okay, starting from you're eating the proteins, right?
Yeah. Okay.
And then protein go to where? Stomach.
Stomach.
Yeah, stomach consists of what kind of enzyme?
Peptin.
Pepsin, yeah, the pepsins.
Yeah.
Yeah, so the pepsin, they will just break down protein, become what?
To become amino acid.
Usually it's become the peptide first, okay, the polypeptide first.
Oh, okay.
Yeah, they will become polypeptide first. Polypeptide, okay. Polypeptide, from the proteins, they become polypeptide. Yeah. So okay, now the acidic chyme, yeah, this one, which is the acidic chyme, they just get into the small intestines. So small intestine, they will have what? They will have another kind of enzyme, which is known as what? The
Trypsin.
trypsin. Yeah, trypsin. Yes, correct. So what the trypsin will do? Trypsin, they can break down polypeptide, become what? Become dipeptide or become amino acids.
Okay.
Yeah, see, from the polypeptide, right? From the polypeptide, they'll become the dipeptide. Okay, dipeptide. Or they become amino acid, amino acid. This is your product, okay?
So the pepsin break protein into polypeptide, and the trypsin break the polypeptide into amino acid.
Yes.
Oh, okay.
Yeah, correct. Yes. Because polypeptide is just very long chain, right? And further cut them down. Right?
Okay.
So this is the chemical digestion, which is the use of the water. Okay, you require use of water and the enzyme to break down the proteins into the small molecule.
Okay.
So you can see there's a pepsin and trypsin here. Yeah, the trypsin here along the way.
Okay.
And then once they become the amino acid, right, they can go into your small intestines.
Okay.
They go into the blood capillary of your small intestines, Monty. Yeah, so that's why small intestines, right, they have the villi, okay? They have a villi. Villi, which is the blood capillary, right? They can absorb the amino acids into the bloodstream. Okay?
Okay.
So later, from the small intestine, they will go to the liver. So what kind of blood vessel?
The hepatic
Portal vein. Yes, well done. So that's why, okay, now, once the amino acid enter to the liver, right, liver, they can do what? The liver, they can do a process called what? Deaminations.
Oh, okay.
Yeah. So what's meant by deaminations? Deamination is like a process, right? You can refer to downside. Okay. I want to show you the deamination process. Oh, what is meant by deaminations? Your school teacher got teach about deamination?
I think I heard of it before, but like I don't think she really taught so much.
Okay, so okay, come. So Monty, have a look on this deamination. Yeah, so deaminations is a kind of process only occur in liver. See, only in the liver, okay? So which is because your body, Monty, if you have excess amino acid in your body, right, your body cannot store excess amino acids. Do you know this?
No.
Yeah, your body cannot store excess amino acids. So if you eat excess amino acid, right, you just excrete out in the urine.
Oh.
Yeah, so that's why the excess amino acid in the body, right, they will undergo deaminations. This is happening in the liver.
Okay.
And then, so deamination is kind of process which is the removal of the amino group from the amino acid, which is this one. You can see, right, this is the amino acid. So Monty, have a look on the amino acid. They just look like this, right? With the C and then the COOH, right? And the H and the R, and then you have this one, NH2. So this NH2 is the amine group, right? Amine group NH2. So this is the one who are being removed away.
Okay.
Yeah, so because of deamination, deamine. See? Deamine. Removal of amine group. Understand?
Okay.
Yeah, so this happen in the liver. So that's why after the amine group, which is NH2, amine is NH2, NH2, okay, NH2. So after the NH2 is being removed, right, they will become NH3 ammonia.
Okay.
And then this ammonia is toxic to the cell, toxic to the body cell.
Okay.
Okay. Yeah. So that's why it's not good for your body also. So that's why your liver, right? The liver, they can convert this ammonia, which is toxic, right, become lesser toxic, which is urea. Urea is less toxic.
Okay.
Even though urea is still a waste substance, you know, urea is a kind of waste substance, but it's still less toxic compared to the ammonia.
Oh, it's like urea, just like urine.
Urea and urine is different thing. Urine is a liquid, okay? Urine is a liquid. Inside the urine, right, they consist of urea. They consist a lot of like mineral ions, salts, ammonium ions, that kind of thing. Understand?
Oh, okay.
Urea is the specific compound.
What compound?
It just look like this. Urea. Let me show you. So it's a product of the like this. See?
Oh.
Okay, it's a kind of colorless, odorless organic compound. See, it's an organic compound. Organic compound is because it's made from what? It's made from hydrogens, nitrogen, and carbon and oxygens.
Okay.
So it's produced in liver. See, Monty, liver produce the urea.
Okay.
And that is the end product of protein breakdown. See, which is when your protein breakdown, right, you'll produce the urea as a waste product.
Okay.
Okay, so this is the thing that you should know. Yeah, it's like compound, okay? Like this. So that's why urea is different from urine. Inside the urine, you have a urea. Understand?
Okay. Is it a organic compound? A compound?
Yes.
Because it's okay.
Yeah, urea is a organic compound because your protein also organic compound, right? Your proteins.
Yeah.
Yeah. So it just like removal of a part of the amine group from the protein. So it's still an organic compound. Yeah, so ammonia is toxic to the cell. You convert the urea, right? Urea is less toxic to the cell. So it's later the urea will be excreted in the urine by the kidney. So you found the urea in the kidney.
Okay.
Okay, so this is the process for the proteins. Okay, so let me show you this picture. Okay, so that's why from the small intestines to the liver, right, Monty? From the small intestines to the liver, so you have what? You have the hepatic portal vein, right?
Okay.
So this hepatic portal vein will go to the liver. So once the amino acid arrive the liver, so liver, okay, they will use the amino acid for what? Assimilation, okay? Assimilation, right? Which is, yeah, the body cell will use amino acid for the tissue repair, growth, or producing enzyme antibody. Okay, those process. Yeah, but what happens if you have excess amino acid? So only the excess amino acid, right? They will undergo deaminations. Understand?
Oh, okay.
Yeah. So they will become ammonia, later become urea. Okay, so that's why urea is being excreted by the urine. See, ammonia is being converted to less toxic urea and then filtered by the kidney for the urine excretions.
Okay.
Yeah. Do you like this picture?
Yeah.
Yeah, make you much more clear, right?
Yeah.
And then how about lipid?
Oh.
Lipid just like this, okay. When you eat a lot of lipid substance, okay, lipid food, the food will consist a lot of lipid, right? So that's why lipid, okay, they will be they just go to the mouth and then to your stomach and then to your intestines, and then they only start their process in the intestines. Understand?
Okay.
Which is duodenum, which is duodenum, okay.
Okay.
So which is at the duodenum part, right? They will just receive secretion from where? From the bile, okay, bile, and also from the pancreatic lipase.
Okay.
So Bao will do the emulsifications, okay? So you can have a look on the upside. Yeah. So Bao, they will do the emulsifications, and the lipase, they will do the chemical digestions. So they'll just break down the lipid, become fatty acid glycerol, which is their monomer.
Can you repeat that?
Okay, which is the lipase, pancreatic lipase, right? They will just break down the lipid, become the fatty acid and glycerol.
Yeah.
Which is the monomer.
Oh, okay, yeah.
Yeah, which is the simplest unit they can directly absorb by the body. Yeah.
Okay.
And so later they will just go into the lacteal. Yeah, see? They go to lacteal, okay? Not blood capillary, they just go to lacteal. Yeah, so lacteal to where? To the lymphatic system. Yeah, so this is the lymphatic system, okay? So just remember, lipid will travel through the lymphatic vessel, and then later the lymphatic system, they still go back to the circulatory system. So they'll just go back to the heart. Okay, but for the exam, right, you just need to know about what? You just need to know about this one, okay? Okay. Just need to know about here. Yeah, like from the lipid, right? See, from the lipid, they'll break down by the enzyme and the bile become the fatty acids and the glycerol. So the fatty acid and glycerol, okay, I forget to add one more word here. Here's the glycerol here. Help me to add. Yeah, so this glycerol plus the fatty acid will be absorbed by the lacteal, okay, absorbed by the lacteal. So they will go to the where? Lymphatic vessel, okay, for the lymphatic vessel. So the lymphatic vessel, right, they just go into the bloodstream and then to the heart and then to the body cell, okay, they will go to the body cell. Yeah, because the heart is the one who will transport the blood to the body cell, right? You agree with me? The heart. Yeah. You just remember that heart will pump the blood to the body cell, and then the heart, when they pump the blood, right, inside the blood, they consist of what? They consist of fatty acids. Correct.
Okay.
Yeah, so that's why they will go to the assimilation stage. Okay? Assimilation stage in the body cell. So that's why the cell will utilize the lipid for energy or membrane formation or cushion for organs. Yeah, which is the usage for the lipid. Yeah.
Okay.
Or the insulator. Yeah, or heat insulator. Yeah. So what happens if you have excess lipid? So if you have excess lipid, right, you can store in your body as adipose tissue. So you have the obesity, or you have arteriosclerosis, which is you will have a narrowing of blood vessel leading to the coronary heart disease. Okay, this one you have deposit. Okay, deposit of cholesterol. Okay, on the wall of the blood vessel, right? So you need to what? You need to the narrowing of the lumen, the blood vessel. So you need to what? You need to the coronary heart disease.
Oh, okay.
Yeah, that's all about the liver.
Okay.
Yeah, okay. I think we discussed about this one. Okay, just have a look at the pathway, and then the end. Okay, so that's why this is some of the common mistake, right, that you need to avoid. You make the mistake. For example, right, you need to know that bile emulsified fat, Monty. You write bile emulsified fats, not digested fat, understand?
Yeah, okay.
Because only lipase will digest the fat. Lipase is an enzyme, bile is not.
Okay.
So and also when you write about the enzyme, okay, like how the enzyme activity is being affected by the change in the pH and temperature, this is the point that you should write. For example, you should say that the enzyme, right, okay, is denatured, okay, by what? Denatured by the extreme pH, right? You can say that what kind of factors? Based on the question, okay, for example, you can say that enzyme is denatured by what? By the extreme pH, or the temperature. Maybe the temperature become too high or too low, it will cause it denature the enzyme structure, right?
Yeah.
So that's why the shape of active site of enzyme will change. So the substrate was unable to bind to the enzyme. Okay, so there will be no enzyme substrate complex form.
Oh, okay.
Okay, and also the digestion pathway. Okay, so a lot of students, they didn't know about what you need to know about, okay, what happens to neutral molecule after they go to the small intestines. So later they'll go to where? You should know about this vessel, okay, which is the hepatic portal veins to the liver. So they will do the assimilations.
Okay.
Yeah, okay, that's all. I think cholera they will not ask. You can skip this one.
Yeah, I think I learned it.
Yeah. And then small intestines, there's pancreatic amylase. Yeah, and then the bowel functions. Yeah, just all those important points that you need to know. Okay, that's all. And then okay, for the misconception, okay, you need to know that actually small intestines, right, they absorb more water compared to large intestine because small intestine is longer, right?
Yeah.
Okay, and then don't think that kidney produces urea. It's the wrong thing. It's the liver who produces urea. Understand?
Okay.
Because only liver they can do the deaminations. Okay, and then bowel is not enzyme, definitely. Yeah. Okay, and then that's all. That's all. Okay. Differentiate the excretion and digestion. Okay. Okay. So other thing you can read by yourself. I'll just show you an example of question, which is the lesson here. Okay. So you know how to answer this part, right? This one. Describe the pathway taken by the protein digestion product from the belly to the liver. So you should say that it's amino acid, right? The protein is being break down into the amino acids. So amino acid will enter to the blood capillary in the belly. So the hepatic portal vein, right? The hepatic portal veins, they will just carry the amino acids, okay, from the intestines to the liver. Yeah.
Okay.
Yeah, the keyword should write the capillary. Okay. Later they will enter the blood plasma, right? And then go to the liver by the hepatic portal veins. This is the keyword, the three things.
Okay.
Okay, this one you just skip first. And then this all ends up, okay, you can just read by yourself. Yeah. All the shape. And then, okay, see what kind of change will affect the pH, what kind of factor that will affect the structure of enzyme. So you can add this one, okay, increase temperature or extreme pH. You cannot write temperature or pH only. You should write increase temperature or extreme pH because this is the one that will cause a structure of enzyme being denatured. Understand?
Okay.
And then this one, okay, physical digestion, okay, the difference for the physical and chemical digestion is physical is only about break down, large becomes small. That's all. Okay, and then the food molecule itself, they don't have chemical change. Understand?
Yeah, okay.
Yeah, so if it's insoluble, they will still remain as insoluble because it's physical, it's not chemical.
Okay.
Yeah, so in order to increase surface area, okay, for the enzyme action later. Yeah, so what's about physical digestion? Include the mouth, okay, the churning action of stomach, or you can write emulsification also. Okay, and then see about the fat digestion, right? Yeah, so you should write about emulsifications and the lipase. So you produce the fatty acid glycerol.
Okay.
Yeah, and then the role of bowel, okay, the same thing. Neutralize. See the keyword, neutralize the acidic kind. Okay, so you create the optimal pH, right, in the small intestines. Yeah, for the trypsin to break down the protein later. So it's not neutral, it's just optimal pH. Understand? The keyword neutralize the acidic kind from the stomach.
Oh, actually, is
Yes?
the hydrochloric acid same as the chyme?
Which one? Hydrochloric acid is same with which one?
Same as the chyme.
Chyme? Okay, different thing. Chyme is what? Chyme is a semi-solid state of the food. So I just describe as chyme, okay? So the chyme, right, is referred to the state of the food, right? Chyme food in the stomach. This one, which is a thick, okay, the thick acidic semi-fluid, semi-fluid, which is just like your porridge, your porridge just a semi-fluid also, right? Yeah. It's referred to the state of the food inside the stomach, but it's different. Okay, the hydrochloric acid is kind of acids. Yeah, the hydrochloric acids, right? Okay, Monty, tell me about the function of hydrochloric acid. Tell me two functions about hydrochloric acid.
It helps break down. It helps make the pH acidic, so the enzyme pepsin can work and is optimum pH. And also it kills the bacteria.
Yes, kill the bacteria. And then first one is what? Create the acid.
Acidic
environment,
pH
right?
or the pepsin.
Yes, well done. You're so good.
Oh, thank you.
Okay. Yeah. Yeah. That's all. That's all about the point for the chapter 7. Okay. So all the best to your exam this Friday. Okay. I hope I make it clear for you. But
Yeah.
you still can read this one. Okay. This one. Okay. This is just now I shared to you. Is about the paper for the recent year, which is from 2023 to 2025. Okay. And then later you can read about this one, which is 2023 before. You can read.
Okay.
You read about the answer, okay? You read about answer. Read about how to write the correct answer with the correct keyword.
Okay.
For example, like proteins, right, is required for the growth and repair of cell, something like that.
Okay.
Yeah. Okay. You can read starting from the last few page first. Read from last page to the front page. Understand? Because last page is about the from the 2023, 2022. Yeah. It's more recent year.
Okay.
Yeah. I think that's all about today. And then I want to show about question, right? The question is a bit nice. I think the question is nice for me. I want to show you the questions. See, all is just the same repeating question only. Where is it? Here. I think it's here. Okay. Yeah, see the physical digestion. Okay, they also describe the physical digestion. See, the bowel emulsify the fats. Yeah, so that's why I use the correct keyword. So IGCSE is just about how you're able to use the correct keyword so you can get the answer. I mean, you can get the mark.
Okay.
Then one thing is about what. So usually they will just combine. They just combine the chapter 4 and 5 us together in the chapter 7 because it's about enzyme.
Okay.
Okay, I want to show about this one. See, this is in I think in 2022. They come out this exam before. 8 mark. Seriously, 8 mark.
Oh.
This is a long essay, okay?
Okay.
Yeah, so see, describe what happened in the carbohydrates in the human body between ingestion to the assimilation in liver. So you need to write the pathway. You need to write the pathway from the point that the starch, right, enters your mouth and then go to the stomach, small intestines until reaching your liver. The pathway, right? So that's why you need to write about what? Mechanical, okay? Mechanical, which is by teeth. Later by what? A chemical, yeah, the chemical, which is to break down the what? Large insoluble becomes small and soluble molecule. So later, which is the enzyme, okay? Enzyme, you should write what kind of enzyme. So for the carbohydrate, right, you should write about the salivary amylase. So you have salivary amylase in your mouth and also in your pancreas, right?
Okay.
Yeah, so that's why they can break down the starch, become the maltose. Later, you have the maltase. You have maltase. Okay, you have maltase produced from where? Where do you produce the maltase?
The epithelial ducts.
Yes, the epithelials in the small intestines. Okay, in the intestines. Yeah, maltase is produced from your intestine cell, right? In your intestine cell, it's different from the amylase, right? Amylase is produced from the mouth and from your pancreas, and then later they'll just go to the small intestines. You get what I mean?
Yeah. Okay.
So they'll just break down maltose to glucose. So the top of bowel again, okay, bowel to neutralize. See the word neutralize, okay, neutralize. So that's why the maltase, right, as I they just require the slightly alkaline pH. This is their preferred pH, around pH 8 to function effectively. So they want to create the optimum pH, for the enzyme to work on.
Okay.
Yeah, see, from the ingestions to the digestions, right? Digestion is about chemical and physical digestions, and then go to what? Go to the absorptions, right? Go to the absorptions, the nutrient absorptions. So now because the nutrient is break down, become the glucose, right? Glucose. So that's why they can be absorbed. See, they can absorb into the blood by the urinary canal through the diffusions of active transport to the villi or microvilli.
Okay.
Yeah, so that's why I tell you just now, which is the glucose, right? Glucose can uptake to your small intestine by two ways, either diffusion or active transport.
Okay.
Yeah, so this is the function of microvilli, which is to increase total surface area for the absorptions. Yeah, okay, so this is the point, okay. Yeah, what happened when your blood glucose level increase, right? The pancreas will secrete insulin. So insulin will just reduce the blood glucose level. So they will convert and become glycogen, okay? So that's why glycogen is stored in the liver. Yeah, and then you talk about aspiration in the liver, okay? So this is the thing you can take note, okay? Aspiration in the liver. So during aspiration, okay, glucose is taken up by the liver and stored in the glycogen. So later, the nutrients, right? The nutrients are assimilated. See the keyword the nutrients are estimated into the body cell and become part of the cell. So they will be used in the cellular aerobic restrictions to release energy. The end.
Okay.
Okay, you actually you don't need to memorize, right? You can imagine the pathway. I mean, imagine your mind, how's the flow, and then you can write the answer already. You just need to know about keyword only.
Okay.
Okay, yeah. So Moti, I hope you're still feeling well.
Yeah. Yeah.
Okay, that's all for today. Okay. So all the best to the exam. So if any question, you can ask me
Thank
again.
you. Okay. Yeah. Yeah. Thank you.
Okay. Bye bye.
Thank you. Bye.